Hey guys, welcome back to the channel. So in this video we'll talk about a new topic which is plant biotechnology. So previously I have made two videos regarding this and I've discussed about agrobacterium-mediated gene transfer. So if you haven't checked it out, you may check it out. So let's start from where we left in the previous video. In this video I'll be explaining the entire process of agrobacterium-mediated gene transfer and each of the steps in descriptive manner. So as you can see, there are as a whole there are 6 steps. So I'll be explaining all the 6 steps in details. So the first step for the agrobacterium mediated transfer of tDNA is signal recognition by agrobacterium. Thereby it attaches to the plant cells, and with that it starts the induction of Vir genes, and with the help of that it produces T strands, and after that we see a transfer of tDNA out of the bacterial cells, and lastly the transfer of tDNA and Vir proteins into the plant cell and nuclear localization occurs. So these are the 6 important steps which I'll be covering in this video. So let's get started. So for the first step, so this is the overall agrobacterium mediated gene transfer, as you can see. So this is the transfer that takes place. So this is the transfer of tDNA. So this is the tDNA from the Ti plasmid, which gets transferred from the agrobacterium to the plant. Alright. So from the agrobacterium it goes and settles down to the plant genome. Alright. So moving on with this. So talk about the first step, which is the signal recognition by agrobacterium. So agrobacterium perceives signals such as phenolics, which can be acetylsyringone or B-hydroxyacetylsyringone, and sugars which are released from the wounded plant cells, and normally these substances are probably part of plant's defense mechanism. So these do act as plant's defense mechanism, and when they release out of the plant, these attract the agrobacterium cells to get attached on them. Alright, so these chemicals or the phenolics released by plants acts as signals which attract the agrobacterium. Alright, so talking about the second step, which is the attachment to the plant cells. So the attachment of agrobacterium to the plant cell is of 2 steps. Alright, so as you can see, so the first step is the initial attachment via polysaccharide, and the second is the mesh of cellulose fiber is produced by the bacterium. Alright, so first is the initial attachment of the agrobacterium via the polysaccharide or the product of ATTR locus, which I'll be explaining in the coming videos, and the second is a mesh of cellulose fiber is produced by the agrobacterium. Alright, also several bacterial chromosomal genes such as CHVA, CHVB, PSYA are involved in the attachment of bacteria to the plant cells. So these are some of the important genes that you might note down for the understanding of agrobacterium mediated gene transfer. So these are the 3 important genes, A, B and A, for the attachment of bacteria to the plant cells. Also CHVA and CHVB helps in the production of cyclic beta-1,2 glycans, and PSCA gene is involved in secreting succinoglycan. So these 2, production of glycans and succinoglycans, are involved in the attachment to the plant cells. Alright, so the attachment to the plant cells. Alright, so these are some of the genes that help in the attachment or proper attachment to the plant cells, so that the transfer of tDNA can be easily done. So coming to the third step. So here comes the most important part, or the induction of Vir genes. So we'll be talking about the Vir genes part as well. So Vir genes. So Vir A. So talking about the first Vir gene, which is the Vir A, or which is a membrane-linked sensor kinase which senses phenolics or the signals secreted by the plants and autophosphorylates. So it all starts with the Vir A gene. So the Vir A gene senses phenolics and thus it autophosphorylates. So with Vir A gene getting autophosphorylated, it phosphorylates and activates with G genes. So I'll be repeating the thing all over again. Alright. So with A genes senses phenolics and it gets autophosphorylates. So in its cells it phosphorylates, and as the phosphorylated with A gene phosphorylating, so this thing is phosphorylating in itself with A genes, it activates with G genes, with G genes. Alright. So as with A gene is getting activated, in it itself activates the other with genes, which now we see is the with G genes, which gets activated due to the autophosphorylation of with A genes. Alright. So now due to this the with G genes gets activated, which thus induces expression of all the vir genes. Alright, so vir genes is a very important part which takes place or which happens for the successful transfer of tDNA. So without the presence of vir genes, the transfer of tDNA is not possible. Alright, so the part of vir genes is very important, and that is why I am trying to explain every part of vir genes. So starts with the with A genes, which also autophosphorylates and thus activates with G genes. And as the activation of with G genes is done, it induces expression of all the with genes, in fact. Yeah. So it induces expression of all the vir genes, and many sugars, but in particular glucose, galactose, and xylose enhance vir gene induction. Alright. So along with vir genes, so there are certain sugars as well. So as you can see, glucose, galactose, and xylose enhance with gene induction. Alright. So it improvises or it betters the activity of vir genes with the help of these sugars. Alright. So these activation of these sugars are not easy. So there's another point for this. This is the last point for induction of vir genes, which says this enhancement requires another chromosomal vir gene, which is CHVE, which encodes galactose or glucose transporter that interacts with vir genes. So as we know now that the induction of vir genes is very important for the transfer of tDNA. So it all starts with vir genes, we're getting autophosphorylated. So with that, it activates with G genes. Alright, so it activates with G genes, and with that, as the activation of G genes is successful, it induces the activity of all the other genes. Alright, so hope these three parts are cleared. So next, the induction of these or all the vir genes can be enhanced with the help of these sugars. So the activity of all the vir genes can be enhanced with the help of glucose, galactose, and xylose. So the activation of or the enhancement of these vir gene induction is done with with the help of these sugars, and with these sugars require another chromosomal genes, which is CHVE genes. Alright, CHVE genes or the chromosomal CHVE genes for the interaction of these sugars with vir genes, so that the induction of all of these vir genes can be done at once. Alright, so this was the point. So talking about the fourth part, which is the T-strand production. So now we just studied about the vir genes part. So now we'll study about the T-strand production. Alright, so the left and the right borders are recognized by endonuclease vir D1 and vir D2 complex. Alright, so as the attachment of Agrobacterium is done with the plant and the activation of vir genes is done, so now what happens is in the TI plasmid, which I have explained in my previous video. So the TI plasmid consists of many parts such as left borders, right borders, tDNA, vir genes, original replications. Alright, so now what happens is the left and the right borders are recognized by endonuclease vir D1 and vir D2 complex. Now, vir D2 produces single-stranded nick in the DNA, and nicking takes place between third and fourth bases of bottom strand of each 24 base pair repeat. Alright, and a nick at the right border 24 base pair repeat provides a priming end, and synthesis of new DNA strand begins from this nick in 5-3 direction. and a new DNA strand displaces the old DNA strand. So I'll be explaining the entire process if it's becoming very much outdated. So yeah, so the left and the right borders are now getting recognized by the endonuclease with D1 and with D2 complex. So now the with D2 produces single stranded nick in the DNA. So it produces nick in the DNA strand so that a new DNA strand can be produced, which will therefore help in the transfer process. Alright. So the nicking helps in the transfer process. So the with D2 produces single stranded nick in the DNA, and the nicking takes place between third and fourth bases of bottom strand of each 24 base pair repeat. So we know that the left and the right border, so the region between the left and the right border consists of 24 base pairs. All right. So the left and the right borders, so this is, let's say this is the left border and this is the right border. In between there are 24 base pairs. All right. So the nicking takes place between third and the fourth bases of the bottom strand of each 24 base pair repeats, and the nick at the right border, pardon. So the nick at the right bottom 24 base pair repeat provides a priming end, and the synthesis of new DNA strand begins in the 5 to 3 direction. All right. So this is the 5 to 3 direction. So new DNA strands starts to get synthesized in the 5 to 3 direction, and a new strand displaces the old tDNA strand. So the old tDNA strand gets replaced by the new DNA strand. All right. And where D2 becomes covalently attached to the 5 end of displaced single stranded DNA. So with D2 is a permanent thing which remains in the entire transfer process. So with D2 becomes covalently attached to the 5 end of the displaced single stranded tDNA. So with D2 remains at its place at the 5 end. All right. And the presence of with D2 makes the 5 end of the T strand less vulnerable to the attack by exonuclease. So it sort of provides protection to the new strand or the T strand. All right. So being present at the 5 end, it also protects the T strand from being attacked by any sort of exonuclease. So talking about the fifth part. So fifth part is the transfer of tDNA out of the bacterial cell. So now the tDNA along with D2. All right. So the tDNA, which is the new synthesized DNA, which has the D2 gene attached at the 5 end complex, is now exported from the bacterial cell by a T pilus, which is a membrane channel secretory system, which is composed of proteins encoded by VID, B operon and VID D4. So the entire transfer is possible with the help of a pilus, which is known as a T pilus. All right. So this DNA, so this tDNA along with VID D2 complex is now moved from the agrobacterium to the plant cell with the help of a T pilus in between. All right, with the help of a T pilus to the plant cell. All right, and now the VID E2 and VID F are also exported from the bacterial cells. So most of the VID genes which are present in the T plasmid or which are present in the agrobacterium are transported from the agrobacterium to the plant cells, so that the localization or the adjustment of the tDNA is done easily in the plant genome. All right. So along with that, so it also contains VIR E2 genes and VIR F genes are also exported from the bacterial cells. So talking about the last step of the transfer. So the transfer of tDNA and VIR proteins into the plant cells and nuclear localization. All right. So the tDNA along with VIR D2 complex and other VIR proteins cross the plant plasma membrane through pilus. All right. So they now move to the plant cells. All right. So they now move into the plant, crossing the plasma membrane through the pilus. All right. Also inside the plant cytoplasm, the tDNA strand becomes covered with VIR E2 protein. All right. And it is now known as the mature T complex. All right. So when it reaches the plant cytoplasm, the tDNA strand becomes covered with the VIR E2 protein. So the tDNA, let's say this is the tDNA strand. It is now covered with the, or it is shelved with the, or it is covered with the VIR E2 proteins. All right. So it is now covered with the VIR E2 proteins, which we collectively call as mature T complex. So this entire part is known as mature T complex. All right. Also, vir D2 and vir E2 proteins contain nuclear localization signals. All right. So these two proteins play a very important role. So vir D2 and E2 proteins helps in the transfer of this DNA, helps in transfer of this tDNA from Agrobacterium to the plant, as well as it contains nuclear localization signals. All right. So through the signal sequence, these protein interact with the plant protein and helps in the transfer of tDNA into the nucleus through nuclear pore complex. All right. So these proteins, so with D2 and E2 proteins interact with the plant protein, all right, and help in the transport of tDNA into the nucleus of the plant through nuclear pore complex. So this is how the tDNA gets settled in the plant after all of the transfer process gets completed. So let's just keep this video till here. Hope you enjoyed this video and stay tuned for more, and thanks for watching this video.